Hi guys, sorry I'm not there today. I just wanted to go over the concept one and the concept two notes, just so you had a little bit more to go off of than just the notes themselves. So concept one is about enzymes and biochemical reactions, which is related to the lab that we just did. So we're going to kind of see how it all kind of comes together and what the heck an enzyme is. So, we talked about metabolism as being something that all characteristics of one of the characteristics of life, so everything that's living has a metabolism or a way for us to make energy. So, in a formal sense, metabolism is all the chemical reactions that we have within a cell. It provides us with energy, so we burn different macromolecules and we get energy from those. And they create really key molecules for us to live and be alive. So, chemical reactions are things that are happening in our bodies. You'll learn a lot about these in chemistry. But basically, in a very simple form, all chemical reactions are things that break and form bonds to make new substances and do what's called create chemical changes. So you might have something like hydrogen and oxygen coming together and forming water. That's a chemical reaction. Or you can have water and it breaks apart and it forms hydrogen and oxygen, also a chemical reaction. And water looks very different from hydrogen and oxygen. Both types of reactions, either breaking or forming bonds, either absorb or release energy. So it takes energy to break a bond. You have to put energy into it. So think about breaking a ruler in half. You have to put energy in order to break that bond. So energy is absorbed by your ruler when you break it in half. If you form a bond, if you put things together, energy is released. So kind of think of that as like that's the way things want to be. They want to be bonded together. So it releases energy because they're now happy to do that. And we have this law called the law of conservation of energy. It's one of the first laws of thermodynamics. There's no loss of energy in the system and no gain of energy. It's just change. So we don't ever lose any energy in this process. But sometimes you can release it or absorb it. And when you release it, sometimes that can be released as heat or light. So if you see heat or light, that's a sign that chemical change is happening. So we have some vocab terms. So chemical reactions in biochemistry can either be, catabolic or anabolic. So catabolic means breaking down larger molecules into simpler compounds that releases energy, or is called exergonic in biology. And anabolic means you're building larger molecules from smaller ones. So that requires energy to do so. And that's endergonic. So all reactions require energy to happen, and there's a specific amount of energy that it takes to start a chemical reaction, and that's called activation energy. So you can think about like when you set something on fire, there's a very specific amount of energy you have to put in to start that fire. That's the activation energy. And we'll see this graph pretty often, and you'll see it again in chemistry. But you have this graph that shows how much energy it takes and how the reaction is progressing. So here you can see you start off with some amount of energy in your environment or your system, and it takes some amount of energy. So it takes uh, this amount of energy, the purple like amount of energy here to start your reaction. So from what the energy you start with to the energy of when your reaction starts, that is your activation energy. And then you can see once your reaction starts, then your reaction progresses further. And so we'll talk also about what it means when you start with more energy than when you finish. So we also have some other terms. I'll move this down here. We have reactants, which are what you start with in your chemical process, and then you have products, which is what you make in your chemical reaction. In biology, we also use this term substrate sometimes to say our reactant. So in the case of the lab that you guys did, the beans were your substrate. Those were the reactants that were going through your chemical reaction. Those are the things that are changed. So at the beginning of your reaction, you have reactants, and at the end of your reaction, you have products. And then we have two types of reactions, endothermic reactions and exothermic reactions. So endothermic reactions absorb energy. So you put heat in or light in. And in this case, like you can start a reaction by heating it up. In photosynthesis, the case is light. So we put light energy in, and the photosynthesis process starts. And that is represented by this graph. So you have some amount of energy to start, and it takes a lot of energy to put into that reaction to get it going. And then when you end, you have more energy than when you started because you put energy into it. So this is an endothermic process because we end with more energy than we started with. In an exothermic reaction, you release energy. So you start with some amount of energy. I'll move this up here, get it out of the way. Start with some amount of energy, and you have an activation energy to get your reaction started. But throughout the process, you release more energy, so you end up with less energy than you started with. An example of this is cellular respiration, when your cells are expending energy or getting rid of energy. You end up with less energy than you started. So the two reactions we just talked about, we'll talk about in more detail later in this unit, but we have photosynthesis and cellular respiration. So this is our chemical reaction for photosynthesis. You may or may not have seen this type of reaction before, but everything on the left side of the arrow are your reactants because that's what's reacting together in the beginning. Everything on the right side of the arrow are your products because that's what you produce in the end of your reaction. And your arrow represents like your reaction happening, and it was progressing forward. So in this case, we have CO2 in water, and you put in light energy. It's stored as chemical energy, and when you create this photosynthesis process, you create sugar on the other side, and you create oxygen. So photosynthesis is how our plants will make oxygen. And so you put that light energy in, which means it's an endothermic reaction. Energy's going in. So remember, we've talked about endocytosis. That's particles being brought into your cell. So endo means into. And then cellular respiration is when you are burning that sugar. So just the process of us breathing in with our oxygen, we're constantly burning calories and sugar, and as a result, we release CO2 and H2O. So the chemical energy in your sugar is converted to chemical energy in our bodies, which is released as ATP, and so it's an exothermic reaction because you're releasing that energy. And so, in our bodies, our chemical reactions or metabolism reactions are controlled by enzymes. So in the case of our lab, our enzyme was our hand, and it's a protein that speeds up biochemical reaction by lowering the activation energy. So it means you need less activation energy to get that process started. Because some of these reactions would happen on their own, but they might be really slow, and we want them to happen faster in our body so we get more energy. So we need something to speed them up. And to do that, we have enzymes, which are also called catalysts because those are substances that will speed up reactions. So there's catalysts in all kinds of different reactions. If you've ever seen elephant toothpaste, they're just adding a catalyst to some hydrogen peroxide and causing that reaction to happen like really rapidly. So that is another example of a catalyst. So enzymes are really special molecules that bind to reactants or your substrate. So our hand was binding to the beans to help break or form bonds and create a new product. So in the case of our lab, our enzyme was grabbing the bean and you're flipping it over to create your new product, which was the bean on the blank side. Enzymes are not changing the reaction, just like your hands stayed the same, and it can be used over and over again. So we just used our enzyme, which was our hand, to do our reaction over and over. And they're really important for life's processes. So here's your enzyme. We have whatever chemical we need to convert. It attaches to our enzyme. The enzyme does the work of changing it into our product. So in the case of the beans, we flipped it over, it was doing the work. And then at the end, you have your products and it releases them, and your enzyme is ready to do that all over again. So you can kind of think it almost like it's like a Ferris wheel process. So like your people go on the Ferris wheel, they get on and they change in some way, and then they get off the Ferris wheel, and then your car is ready to go again. They are very specific for what they're trying to accomplish. They have an active site that only fits one type of reactant. So our enzymes are really specialized to do one job. And once the substrate or the reactant connects to your enzyme, it binds really tight. And this is called an induced fit. So if this was your enzyme, this is the active site, and it's ready to bond to whatever your reactant is, and it fits really well in there. So here's another example. Let's say this was your substrate or your reactant. This is your active site, ready for it to bind or make an induced fit. And then in the process, the enzyme turns it into its product, and it releases the products out into your body. And it can break bonds in a substrate to form two products. So in this case, like if this was your substrate and it was combined, it could break them apart into two separate things, which would be these products. But it can also do the opposite. It can form bonds to make one product. So let's say you have two reactants or substrates, and then it meets in your active site. The enzyme helps bond it and releases it out into your body as a product. Denaturation, which is what we also modeled with our lab, is when an active site gets deformed and loses its shape, which means your biological activity is going to slow down because you weren't able to go through your chemical reaction like you're supposed to. So this was a case where we taped our hand into a 4. It was deformed, and we could still kind of get things done, but it was slower. So this could be due to different environmental changes like pH or temperature change or ion strength. And some enzymes can be renatured to their original shape, but not always. So if this was your normal enzyme, let's move this out of the way. If it got denatured, it would just be like a different shape or something like that. And then you'll notice, like before, we had the very specific fit for our substrate, and now it's not necessarily the same. So it's not going to work as well as it did before. And then we know we have some ways we can change the rate of a chemical reaction or change how fast it goes. Temperature increases can always increase the rate of a temperature of a reaction. So also you can think about what's a good example? If you think about like dissolving sugar, increasing the temperature of your water increases the ability of you to be able to dissolve. Your molecules are moving faster, colliding more, so your reaction will happen more rapidly. If you change the pH of your system, it can change how rapidly you can go through a reaction. So, like how acidic a solution is will affect how your enzymes work. Other examples, and these are just a few examples, there are definitely other ways to do it, but you can change the concentration of your reactants or your substrate so that the more of it you have, the more your reaction will progress forward. So you can think about if you had even more beans, you would have been able to kind of use them all. If we had unlimited time, you'd be able to do the reaction more. If you had a catalyst, like another enzyme, so that would be having your coenzyme. So now you have two people able to go through your reaction. You're speeding it up, and you have more of your reaction that's going to be completed. And then the last one we modeled was the competitive inhibitor, which was competing for the active site on the enzyme. So if this is your enzyme, instead of being able to pick up beans, you have a tennis ball that wants to be attached to your enzyme, and that's going to compete with your uh, beans for being able to access the enzyme to undergo your chemical reaction. Okay, and then I'll do another video for concept 2 in case you need to take a break.